The endospore stain, using the Schaefer-Fulton method. To begin the endospore stain, you must first prepare your slide. Label the slide and then draw two circles, one labeled 24 hours and the second labeled 1 week. You will be comparing the formation of endospores between a 24-hour-old culture of bacillus and a 1-week-old culture of bacillus. After you have labeled your slide, you will transfer 1 drop of clean deionized water into the 24-hour circle and 1 drop of clean deionized water into the 1-week circle. The water will be used to mix the bacteria onto the slide. Your loop should be sterile for the transfer of the water. Bacillus is a gram-positive endospore-forming rod. Another genre of endospore-forming rods that are gram-positive are the Clostridium. Both Bacillus and Clostridium bacteria are responsible for several pathogens, including anthrax, tetanus, botulism, and gaseous gangrene. Aseptically obtain some bacteria from the 24-hour-old culture. You will mix this into the circle with the water that is labeled 24 hours. This process will then be repeated for the 1-week-old culture. Be sure to sterilize your loop in between each transfer. Bacillus and Clostridium both form endospores when environmental conditions become unfavorable. As you would suspect, the 1-week-old culture will contain a large amount of spores, or endospores. Endospores are composed primarily of keratin, calcium, other proteins, and acids. Peptidoglycan is also present in the spore coat. This makes endospores very tough and resistant to desiccation, dehydration, destruction by disinfectants, or other chemicals. Once the bacteria has been smeared on your slide, you need to let it air dry. After air drying, the primary stain, known as malachite green, will be applied over moist heat. Here, a beaker has been placed onto a hot plate and turned on until the water is steaming. The slide is placed on top of a wire rack, so it is exposed to the steam from the beaker. A small piece of paper towel the size of the slide is laid down on top of the slide to help adhere the stain to the slide and prevent it from drying out. You will apply the malachite green and allow it to sit over moist heat with the primary stain on it for approximately 7 minutes. Every 1 to 2 minutes you will need to check the slide to make sure that the stain is not drying out. Here, the stain is no longer shiny and the paper towel is somewhat visible. It is at this point that you should reapply a few drops of the malachite green in order to keep the slide moist. Once 7 minutes has gone by, the slide should be removed from the heat and placed on a staining rack. After removing the slide and placing it on the staining rack, allow it to cool for a few seconds. At this time, you may also remove the paper towel. The malachite green will be rinsed off with water. In this case, water is acting as a decolorizing agent. Once the endospores have cooled, they will tighten up. This will cause retention of the primary stain, malachite green. The water is used to remove the malachite green from vegetative cells. Those are cells that have not yet formed endospores and are metabolically active. Malachite green as a primary stain is highly soluble in water, so it is easily rinsed. After rinsing with water, put the slide back on the staining rack. You are now ready for application of the counterstain. The counterstain used in the Schaefer-Fulton method is safranin. Safranin will stain vegetative cells and endospore-forming cells red. Safranin should be allowed to remain on the slide for 1 to 2 minutes. After 1 to 2 minutes, the safranin is then rinsed with water. Safranin is a basic stain, so it has a positive charge, and it will adhere to the vegetative bacterial cells. This will cause the vegetative cells to stain red, endospores themselves to be green, and free spores or mature endospores to also be green. After rinsing, dry the slide with bibulous paper. It should be observed under the microscope using the 100x oil immersion objective. Shown here is an endospore slide under the microscope. On the left, you notice a free spore. Free spores are mature endospores in which the vegetative portion of the parent cell has deteriorated and fallen off. At this point, in a free spore form, the bacteria are dormant or not metabolically active. At the top right is a vegetative cell. Vegetative cells are cells that remain metabolically active and have not yet formed endospores. In the middle right, you see an actual endospore. This is a combination of a vegetative cell with a spore in the middle of it. This cell is most likely in the process of sporulation. Sporulation is the formation of an endospore inside of the bacteria cell. Endospores are not a form of glycocalyx. They are a dormant form of the bacteria formed in response to harsh or unfavorable environmental conditions. They provide long-term storage of water, nutrients, they prevent dehydration, and they also prevent destruction by chemical or physical methods. Endospores make the bacteria resistant to UV irradiation as well. Here is a picture of the 1-week-old bacillus culture. Notice that in this picture there are many more spores than there are vegetative cells, and there are very few, if any, actual endospores. This is due to the age of the culture. Again, this culture is 1 week old. The endospore stain using the Schaefer-Fulton method is a very simple and easy method to complete, even though there are several steps. The endospore stain is a structural stain and is often used for differentiation and detection of gram-positive endospore-forming rods.